The sequence of nucleotides in our DNA informs us of the alleles we have and therefore with high probability the traits we show. Sequencing involves obtaining the order of nucleotides. Obtaining the sequence of one of the DNA strands of the double-stranded DNA is sufficient as the other strand's sequence can be determined being complementary. If we sequence both strands and find one is not complementary to the other, then there is probably a sequencing error in one of the strands. Errors do happen, and that's why repeating procedures is so important. One approach to sequencing is sequencing by synthesis, such as used in automated Sanger method or Illumina's next-generation method. As sequencing is carried out by DNA synthesis, its requirements as for DNA synthesis are a DNA primer to initiate the replication, DNA polymerase to link nucleotides, and the 4-deoxynucleotide triphosphates, DNTPs. The synthesis process begins with the annealing of the primer. The problem is we do not know the sequence of the template to determine a complementary primer, but the problem has been solved. If the sequence expresses a protein, reverse transcribing the gene based on the genetic code can provide degenerate primers. Otherwise, the unsequenced DNA fragment could be attached to a vector or adapters whose sequences are known, enabling primers to be designed. And if the fragment to be sequenced is from a previously sequenced species or previously sequenced related species, the conserved sequence regions can be used to design primers or a series of degenerate primers. Automated Sanger sequencing requires a low concentration of dye-labeled dideoxynucleotide triphosphates, DDNTPs, in addition to the standard requirements for in vitro DNA synthesis. DDNTP, unlike DNTP, does not have a hydroxyl group on the 3' carbon and cannot form the phosphodiester bond between nucleotides. So once incorporated into the DNA strand, will stop replication. In the automated Sanger sequencing, the 4-chain terminating dideoxynucleotides are dye-labeled with a different colored fluorescent label to detect the nucleotide added at a specific position. Having designed the primers, running the sequencing reaction begins with heating to denature the DNA double helix that is separating the two DNA strands by breaking the hydrogen bonds between the bases of the complementary strands. The heating is followed by cooling to allow for annealing of the primer at the 3' ends of complementary strands as DNA polymerase reads from the 3 to 5' and builds from the 5 to 3'. Once the primer has bound, the temperature is raised slightly to allow DNA polymerase to add nucleotides, extending the strand complementary to the template strand. The nucleotide joined to the strand being synthesized may be DNTP or the dye-labeled DDNTP. Since only small amounts of DDNTP are included in each reaction, DNA polymerase will more often add DNTPs to the chain until by chance the polymerase adds a DDNTP. Once a DDNTP is added, no further nucleotides can be added, so the strand will end with a DDNTP. The process of heating, cooling, and heating to allow for denaturation, annealing, and extension for DNA synthesis is repeated for several cycles as set on the sequencer. By the time the cycling is complete, the termination of DNA synthesis on the occasional addition of a DDNTP leads to the generation of numerous copies of the DNA sequence of all lengths spanning the amplified fragment. That means each reaction now has a set of DNA fragments with the same starting point but different endpoints. The ends of the fragments will have dye-labeled DDNTPs, which can indicate the nucleotide at a specific position. These oligonucleotides are size-separated using electrophoresis. As smaller fragments travel faster, the nucleotide nearest the primer is first detected, followed by the detection of the terminal nucleotides of fragments of increasing size by the nucleotide's characteristic fluorescence. The data recorded by the detector consists of a series of peaks in fluorescent intensity called a chromatogram, from which the DNA sequence is read. As nucleotides are being added at the 3' end, the sequence from the first nucleotide added to the last nucleotide added is 5' to 3'. The sequence read is the sequence of the newly synthesized DNA strand, so the sequence of the template strand will be complementary to the sequence read and antiparallel. Next-generation sequencing by synthesis technology is similar to Sanger sequencing, in which DNA polymerase catalyzes the incorporation of nucleotides, extending DNA on the primer, complementary to a DNA template strand. But the nucleotides used are modified with a fluorescent labeled and DNA extension blocking group, which can be removed. In next-generation sequencing by synthesis, a library is prepared where DNA samples are extracted from cells, purified, fragmented by high-frequency sound waves or enzymes, and the short DNA fragments ligated at their ends to sequences of DNA called adapters. These adapters consist of nucleotide sequences for the DNA fragments to hybridize to the oligonucleotides on the flow cell and be sequenced by allowing primers to anneal. The adapters also have barcode sequences to identify specific DNA fragments, so many different DNA fragments can be sequenced simultaneously on the flow cell, leading to this method being referred to as massively parallel sequencing. The library created is then denatured to form single DNA strands. It is added to the flow cell, which has anchored all over it oligonucleotides complementary to the adapters on the DNA library. The DNA fragments of the library hybridize to the oligonucleotides on the flow cell and are replicated, then denatured, and the strands not bound to the flow cell are washed away. The DNA fragments bound to the flow cell undergo bridge amplification to generate clusters of the same DNA. Cluster generation is necessary so that sequencing produces fluorescent signals high enough in intensity to be detected. In bridge amplification, the DNA fragment captured by one of the oligos on the flow cell folds over. It anneals to the second oligonucleotide sequence to form a bridge and is then replicated. After replication, the strands are denatured, creating two single strands. Each single strand forms a bridge and is then replicated and denatured repeatedly, creating localized clusters of both forward and reverse strands attached to the flow cell. To prevent the sequencing process from picking up different signals from forward and reverse strands, only one strand is sequenced at a time. So one strand, say the reverse, is cut and washed away, leaving the forward strand ready for sequencing. In sequencing, primers added bind to the adapters at the top of the DNA strand. DNA polymerase uses one of the four fluorescent terminator-attached nucleotide bases which has been added to extend the DNA strand. Each fluorescent terminator-attached nucleotide base emits a unique fluorescence when excited, and so whether an A, T, G, or C base is at a specific location on the sequence can be identified. The complementary nucleotide anneals to the target and is ligated to the primer. The nucleotides not incorporated are washed away and the flow cell is imaged. The fluorescent terminator branch is then cleaved and removed, allowing further strand extension. Subsequent addition of nucleotides, followed by removal of unbound nucleotides, imaging, cleavage, and fluorescent terminator branch removal is repeated many times, and thus the sequence of the strands are obtained. Once forward sequencing is complete, the read sequences are denatured and washed away. The barcodes on the adapter identifying the sequences and the reverse strand sequences can also be read. In both Sanger and next-generation sequencing, bad reads are filtered out, These include reads with low-intensity fluorescence or overlaps of different colored fluorescence due to presence of different sequences together. Sequencing can only order a short stretch of nucleotides, say around 2000 bases in Sanger sequencing and smaller in NGS sequencing by synthesis. But even the smallest human chromosome is 48 million base pairs, so there are approaches to obtaining long nucleotide sequences. The shotgun approach can be taken where copies of the same DNA are cut randomly and thus fragmented at different positions along the DNA strand. The jumble of fragments once sequenced are put together in the correct order to obtain the continuous sequence or contig. Two alternate methods may be used. To form contigs, when an organism's DNA is sequenced for the first time, the fragments are compared and assembled based on overlaps. Overlaps occur due to the same DNA being broken at different positions. To form contigs, when a member of the same or closely related species has been sequenced, the DNA reads produced are aligned or compared to the reference sequence and placed alongside their identical or most similar counterparts. One may also use primer walking to obtain long sequences. In primer walking, a series of Sanger sequencing reactions are used. Based on the sequence of an initial fragment, primers are designed near one end to amplify the sequence further along the strand. Then this sequence is used to design a primer near its end and sequence even further along the DNA strand. And so repeating this process many times reads a long continuous sequence. So that's sequencing in brief. Let's test understanding of these concepts with a few questions. Question 1. What would happen in an in vitro Sanger sequencing mixture containing the 4 ddNTPs and no dNTPs? A. No new phosphodiester bonds would be formed. B. All the products would be 1 nucleotide longer than the primer. C. All the products would be 4 nucleotides longer than the primer. D. Products of various sizes would be synthesized. E. All the products would have the same size as the template strands. The answer is B. All the products would be 1 nucleotide longer than the primer. Only one of the ddNTPs can be complementary to the base located just beyond the primer and bind to the primer. Since a ddNTP is binding, further extension is not possible. Question 2. You sequence gene Z and observe the following pattern for the coding or mRNA-like region corresponding to amino acids 1 to 6 of the protein. A. Label the DNA's 5' and 3' ends by filling in the boxes and show the direction of DNA strand synthesis by an arrow between the boxes. B. Write the sequence of the DNA strand that was used as the template for DNA sequencing. The answer to A is the shortest fragment has to be the 5' end and the longest at the 3' end as nucleotides are added to the 3' end of the primer. The answer to B is 3' ATT CT GAAT CC GATA CTT 5' because the template strand is complementary and antiparallel to the DNA strand sequenced. Question 3. Next-generation sequencing allows the simultaneous sequencing of millions of different DNA molecules. Shown is a very simplified representation of the data produced from Illumina high-throughput sequencing. You have 5 DNA samples to be sequenced, 1, 2, 3, 4, and 5. Primers are attached, and the appropriate reactions are performed. What are the 4 bases in the strand replicated from sample 3 if the nucleotides are fluorescently labeled as shown? The answer is that the strand replicated has the sequence 5' AACT3' based on the images of the fluorescence after every cycle of nucleotide addition. Question 4. In the context of DNA sequencing, what is a contig? A. A set of overlapping DNA fragments. B. The number of nucleotides per centimorgan. C. The amount of DNA sequence that is found in one read. D. A mature mRNA in which introns have been removed. The answer is A. A set of overlapping DNA fragments, as contig refers to contiguous, which means continuous. Question 5. State whether the following is true or false. Primer walking employs a series of Sanger sequencing reactions where primers are designed near one end of each sequence obtained to enable sequencing of the region further along the DNA strand, thus obtaining a long continuous sequence. The answer to this question is true. I hope this video was useful in understanding some of the concepts involved in nucleotide sequencing. If it helped you and you would like to see other video tutorials on biology, do look up this channel and subscribe to it. Thank you.